welcome everybody to the first of these in 2026. I've slightly lost track of how many years we've done these, but it's a great pleasure to start off 2026 with one of my dear old friends. I probably shouldn't say old, but dear friends, because we're both young men. Normally, as you know, we do this at 4 o'clock in the UK time, which would make Simon doing this at 3 a.m. in Sydney time, which is where Sydney is. So we thought we would make it a bit later. So thank you very much for joining us at this slightly unusual time. As Eli says, the idea of the WPC is to bring together all the different stakeholders. And this is a wonderful opportunity that we have through these research spotlights to talk to someone who's made a major contribution to Parkinson's research, talk a little bit about their life, how they got into Parkinson's, how they got into their area of research, and then delve in a bit to their area of research. Obviously, if you have questions, as Eli said, do put them in the chat so we can see what these questions are. But it's great to welcome Simon Lewis. So Simon is a neurologist in Australia, but the moment Simon starts speaking, you will probably twig that he's not actually from Australia. So it's great to have you join us, Simon, and you're very welcome. So perhaps I could start off by just saying, so you just explain what you do at the moment. So you're in Sydney, where it's quite warm, and you're a professor of neurology at one of the universities there.
Yeah, that's great, and thanks everybody for joining or watching this on delay. And it's a great pleasure to be working with the World Parkinson's Coalition, and a great job that you guys are all doing. And I hope that everyone is looking forward to the meeting later this year, I think, in Phoenix. I'm sure you'll plug it later. So essentially I'm a neurologist who did my training back in the UK, and moved over to Australia. It'll be 19 years later in February, really to explore opportunities and options that were here. And in that time, I've worked in the public hospital system, and also as a clinical academic, largely at the University of Sydney. But about 2 years ago, my team were approached about a move to a younger university, Macquarie University, and they have a lot of vision, a lot of great ideas about how we can put basic clinical scientists, the sort of people who deal with, blood biomarkers, and clever people who do processing of data, and clinicians together. And so we're hoping it'll be about the middle of this year when we'll open a new centre that will specialise not only in Parkinson's but other degenerative diseases of the brain. So very exciting times.
Very exciting time. Lovely weather as well. And I was there. I was fortunate enough to be there last year, and it's a wonderful place. I have to say, it's got amazing energy. So Simon, let's go back to the beginnings of your life, if you can remember that far back, and where you grew up, and what got you into medicine in the first place. I mean, do you come from a very medical family, or?
No, I certainly don't. I'm the first person in my family to go to university. My father, I got him into doing genealogy at some point to try and get him interested in computers and the internet. And the bottom line is that he was very interested in the idea of finding out our family tree. And of course, it was a waste of time because we're actually from South Wales, so everybody was a miner. They'd be all coal miners. So essentially, even the wives, the children, everyone was a miner going back 5, 10 generations. And so it was a bit of a surprise. And I guess I was very motivated from an early age to get involved. And I think when I talk to people who are interested in doing medicine, I often sort of ask them what they want to be when they grow up. And I think what you really want to hear is people who say, it's a calling, it's what I feel like I really want to do. Most people don't know what they'll end up doing when they go to university and end up doing medicine. Certainly, I don't think I did. I had an idea then I'd be a surgeon, a trauma surgeon, and because I thought that'd be exciting. And actually, I think it's probably the least exciting thing, although it sounds very wow cavalier. But actually, it turns out that I think people like ourselves, Roger, who end up in neurology, really much prefer the mental challenge and trying to live with uncertainty. And so for me, getting into medicine was a challenge, but trained at the University of Wales College of Medicine, so in Cardiff, and then came out with my training and worked along the South Wales corridor across sort of Cardiff and Newport and Bridgend, those sort of bigger centres, before really thinking, okay, well, I tried a lot of specialties. I kissed a lot of frogs. So, I did the cardiology, the chest medicine, and kidney medicine. I
Yeah,
did a lot of different specialties. I was really quite good. I could put a central line in and a pacemaker and all of those things, but it
yeah,
wasn't very exciting and interesting. And I remember going to my first neurology clinic, and I spent half a day in the neurology clinic. I think I saw about four patients. And at the end of it, I had no idea what was going on with the majority of them and realized this is for me. I have no idea what this is. About 3 years ago now, the Medical Journal of Australia asked me to do one of these things about your career, and the
yeah,
title was The Case for Uncertainty. And I think what neurology especially does is it goes, well, we can't take that organ out and look at it like the cardiologists
yeah,
do or replace it with something from a pig. Our game is often living with that uncertainty and say, well, on the balance probability, I think it's this, and I think we're going to try this, and if it doesn't work, we're going to try that. And so it's a very different specialty, I think.
Okay, so you were sort of attracted to medicine because this is quite an exciting area to explore. Not quite sure where you'd end up in medicine, but even in those early jobs you were doing, you could see that neurology held an attraction. As you say, it's the history, it's the examination, it's sort of clinical, picking up clinical clues and then working on that hypothesis. So I think we first met. I'm going to have a wild guess here, but it was. I hate to say it was the last century, but it probably was. Uh, and and at that point when we first met, and we were very fortunate for you to come and do research with us. Um, had you decided at that point that neurology actually was going to be what you were going to do, and you were going to research Parkinson's, or was it more? I quite like neurology. Let's give it a bit more of a go, and I'll have a go at research, and if it works out, okay. If not, let's move on.
The bottom line is, and most people who are watching may not know the system, but essentially I was very committed. I knew that I wanted to do neurology, but at the time you couldn't get into neurology training without having done some research, a higher degree, and basically you're kind of at the mercy of what jobs come up. And as it happened, before the interview in Cambridge, there was a possibility of doing motor neurone disease in London, but I was overseas and they wouldn't hold off on the interview. And but for that, I may have ended up doing motor
Oh,
neurone. And I often say to people, it really doesn't matter what you get into because you'll get into it and it'll be fascinating.
yeah,
And the truth is that your job, the job that I took, was extremely exciting. And I remember us talking, it was 1999, and I remember asking you about your program. I did my due diligence. I wanted to know about you, I wanted to know what you were thinking. And you had this sort of cradle to the grave approach where you said, I want to know how the disease starts, and I've got this post-mortem thinking, I want to put it all together. But you also had this other idea, which was to say, well, as I've just mentioned, you can't take the brain out in real life and study it, but you basically were advertising this new technique at the time with functional MRI, where instead of just lying in the scanner and us taking pictures of the structure of your brain, you could lie in the scanner and actually do things, whether it was cognitive tasks or visual tests or something, and the brain would get activated and you could work out which parts of the brain were doing the thing. And of
yeah.
course, in disease states, work out which parts of the brain weren't working so well. And that would help you understand the underlying problem, the neurobiology, which hopefully guides you towards better treatment. And I thought that was absolutely, I mean, it was the biggest draw I thought of your role. It was just fantastic.
So you were really drawn into research, not necessarily because there was a family history of Parkinson's, or it was more that you were drawn into research. You liked the research program, and you thought, I quite like these new tools, this new imaging. We can actually look at the function of the brain. So you started in Cambridge doing your research at that time, and that was over a number of years. And I mean, if I remember correctly, and I'm of an age where remembering correctly isn't always correct, but you were looking, we were looking at subtypes of disease. We were trying to understand, the different forms of Parkinson's, which in those days, which is 25 years ago now or more, was a sort of relatively new concept. This idea of what we call heterogeneity, different types of disease. And when you started doing research at that time, did you think, actually, this is a bit of a disappointment? I thought it was going to be very exciting, but it's a bit boring because there's a lot of tedious stuff I need to do here. Or did you think, actually, I think I found my niche doing research, doing imaging, Parkinson's. Actually, I can see this is my passion.
And I guess the bottom line is I think that you have a yearbook for all of your former students, and the entry under my yearbook probably reads least likely to ever have a clinical academic career in the future, and I think that was probably fair. As I mentioned earlier, it was a means of getting into a training program, and I thought, well,
Mhm.
this is very exciting. And I must confess that I talk to students now about the research that they do, and I talk about a thing called the support group challenge. And Roger, you really introduced me to support groups where, in the UK, obviously, the network of usually regional support groups, and this is kind of a support group type framework where people like you and I would go along and we would present the work that we were doing. And importantly, that work was often funded by the people sitting in the room. And what I say to the students now is, okay, so at the end of that presentation, what do you want the people in the audience to think or say? Because if they look at you and go,
Yeah.
um, I didn't understand that thing about the protein, or there was that thing about, and and really we as educators and researchers need to be able to understand the problems that we see in the clinic from the perspective of the patient and their family, the
Mm.
caregivers, and say, okay, this is the research we're doing. This is why we're doing it, because in actual fact, if we understand this bit, we might have a better approach for that bit. And I think that's where some of that excitement. But at the end of my time in Cambridge, it was very much, well, let's get into a clinical training program. I've got my ticket now, and I'll just swan off and be a neurologist. And I've had a little contribution, and the world will forget me so quickly.
I don't think people will forget you very quickly, Simon. But you
No.
you elected not to do that. So you elected at the end of your period of research to actually stay in research and for a while to stay in Cambridge. And and it was around this time, I think you started, having done this sort of rather seminal work, I would say, in heterogeneity, that you got quite interested in gait, in walking in Parkinson's, which, for many on this call will know this is a major problem, these issues where, yeah, I mean, obviously people when they begin have slightly smaller steps, don't swing their arms, but then you get this sort of problem where your feet get stuck and you get freezing and such like. So how did you get into that? Because initially you were just fascinated by scanners, Parkinson's, subtypes of disease. So what swung you into looking at gait? And then I want to discuss, why did you then decide to run thousands of miles away to take that project?
Yeah. So, I mean, I guess just to bring it back, the work that we did together was very much focused on, okay, what subgroups are there? But it had a real flavor of cognition. And then, why are some people really good and some people really bad in terms of their cognition, and what pattern of cognition? And one of the things that I'm talking to medical students and trying to explain is that when we do a wiring diagram of the brain, and we say, well, this is the circuit that makes your arm move, that's motor. And then this is the circuit that makes you think, and that's cognitive. And this is the circuit that protects you from danger. And we call that, I don't know, a limbic circuit or a salience, what's going on in my environment. And the fact of the matter is, monkeys swing through trees by swinging their arms, looking for food and trying not to get eaten. And they have to put all of that together. And to me, the idea of saying, well, okay, what is it that actually allows us to put everything together, and where would it fall down the most? And if you look at humans, as you do a lot, the most complicated thing we do as humans is talk. So here we are talking.
Mm.
The second most complicated thing we do is walk on 2 feet. And of
Yeah.
course, we walk on 2 feet and talk. And it struck me that, when you're walking on 2 feet, talking and worried about falling over, this is that perfect monkey model to say, okay, well, this is a real challenge. How do we see these things overlapping? And rather than looking and going, well, the feet moving, that must be motor. But actually, I've got to judge the distance to the doorway and the width of the doorway, and am I going to fall over the dog? And all of those things are really important. And I remember the very first patient that I saw when I was doing the research, and if you remember, went on the road and I saw 200 patients in about 4 or 5 months, visiting people in their own homes, which is absolutely one of the best things I've ever done. Because when people come to the clinic, they're polished, they're best dressed. The wife will say, "Well, he never walks like that at home," and it's just not the real product. And I remember visiting this gentleman out in Norfolk, and he had one of those frosted glass front doors. And I rang the bell and I could see this shadowy figure coming up the corridor, and then it disappeared. And then I saw it again, and it came a bit, and it disappeared. And by the time I opened the door, it happened about 3 or 4 times. And as we walked in, he basically had a freezing episode where his feet stuck to the ground, then collapsed to the ground. And I was like, "Oh my God, are you okay?" He said, "Oh yeah, that happens all the time." I'm like, "What?" So it struck me that we had this problem that was real. And interesting, some of the research we've done now have, suggested how important gait is because, of course, it's linked to independence. And so it struck me as being a real problem that we didn't have many solutions to.
Yeah, and I think that was very prescient of you to sort of pick that as a research topic. I suppose freezing of gait and problems with walking is well recognised Parkinson's disease, as I said, and freezing of gait, dare I say, even today is disabling and difficult to treat. But one of the problems, of course, is how do you study it? Because at the turn of the century now we have lots of devices where you can measure things, but of course 20 odd years ago there weren't many devices for measuring this. So you came to studying gait, freezing of gait problems with walking, which are clearly very important for independence. And I'm sure all of us, even if we haven't had Parkinson's, when you've injured your knee, you've injured your hip, you've injured your ankle, you realize how dependent you are on walking and how difficult it is. And so you decided this would be something to be interesting to study. So how did you think about doing that? Because you're obviously like scanners, but you can't walk in a scanner, really. Well, I'm sure you can walk in a scanner, but you won't get a useful picture. So the question is, how could you take something which is uniquely human? So you couldn't, you could obviously look at it in animals, but they're quadrupeds. So and freezing is such a unique thing to this condition. So how did you think about how you could study this in people?
So if people are brave enough to have a read of the blog, they'll actually see the true story. So I remember I'm in a pub in London, or a cafe, I think it was, with Adrian Owen, who Adrian Owen is one of the big doyens of functional imaging with brain scanners. He's sort of one of the guys that's led this issue of consciousness. What does it mean to be conscious? Looking at patients with permanent vegetative state now in Canada, doing his research, and obviously a good friend of yours, Roger. And we were debriefing about my research career up to that date, and I was saying how I didn't think it would really achieve much. And he kind of said, well, why? And I said, well, it's cognition, and I don't know that we're. And he said, well, what? And I said, well, we should do something about this gait thing. And he said, well, how are you going to do that then, wise guy? And I got very angry on the train ride home to Cambridge. And I was thinking, well, how would you do that? Now, of course, we have a lot more techniques that we can talk about later on. But at the time with the functional MRI, it struck me that maybe we could trick the brain by using virtual reality and really having a go where in a scanner, we're very confined. There's a tiny little mirror that projects an image so that you can see it. And you can do things with buttons with your hands or at pedals with your feet. And so it occurred to me that perhaps if we could get patients in the scanner, immerse them in a three-dimensional virtual reality environment where they're looking at it as though they're in the first person, then perhaps we could trick the brain. And the problem I had is I can't program any computer stuff. But I've realized that I didn't necessarily need to be able to do that because there was a whole industry out there of what they call shoot 'em up games where people can walk around in an arcade killing aliens. And it struck me that people had already worked out how you could walk through a three-dimensional environment, round corridors and shooting aliens. And if I could get rid of the shooting aliens part and capture, some of the other triggers like cognition, like threat, anxiety within that environment, and then be able to record the timing of it and then lock that to what the scanner was seeing in the brain, then maybe that would be a way of exploiting imaging to look at a process like gait. It's a surrogate, but it seemed like a good idea at the time.
Yeah. Well, I think it's an excellent idea, and it's obviously proven to be a very useful area. And obviously, virtual reality was a relatively new thing in those days. So if we just wind back a bit and think, so freezing of gait is very characteristic in narrow spaces, doorways, and such like. So before we come on to what you've discovered in the imaging and then how you've taken it on and moved it to Australia, what do you think is the problem? Why do people freeze? What is the fundamental problem? Is it a sort of overload and you just switch the program off, or what causes it?
Yeah, so and it's interesting because that was the next thought process, which was, okay, do we have a hypothesis that we could
Mm.
then test if we had a paradigm? And in actual fact, the hypothesis paper was written before the paradigm was ready. And the hypothesis paper said, look, if we have these motor, so movement and cognitive thinking and anxiety or salience or limbic all going, is there a point that they all have to cross over? A bit like a telephone switchboard, where the switchboard operator basically becomes overloaded and goes, well, people on this call may be old enough to understand the concept of a switchboard operator. I think if I was speaking to,
Yeah.
the next generation, they'd be lost. And it struck me that, with a disease like Parkinson's, you then also say, well, are there any, if you like, pressure points? And you go, well, it's a dopamine depleting disease. So is there a pressure point where those things would have to be processed in parallel that would depend heavily on dopamine, especially as we know that freezing occurs in later stages of disease more frequently and when patients are wearing off and their medications aren't. And so that puts you into the basal ganglia, the switchboard of the brain. And then you say, well, okay, well, if you did overload the system, what would happen? And the answer is you get this massive output that inhibits movement, which is
Okay.
presumably a safety first movement because essentially we're not supposed to be continuously moving. We're not amoeba. And so we have this situation where you say, well, the default program says don't move, you might fall. And then you're allowed to move because you facilitate things. But effectively, in a situation where you're overloading the system, and of course, you've mentioned narrow spaces and turning being common triggers, that would then allow this parallel processing to fall over. And that allowed us then to think about, well, okay, if you did a virtual reality environment and you could pick periods where the system was falling over versus where it was working, you could compare the two brain scans and say, okay, well, does that fit the hypothesis? Because
Mm.
essentially science is hypothesis driven. So you basically need a hypothesis to test it and then, like a straw man, tear it apart and say, well, that was rubbish. Or actually, that worked. What else did that allow us to do next?
So the idea would be that when you're going through certain situations, and I can imagine in a narrow space, you've got a lot of information coming in, you're having to think a lot more. The idea is that all comes in, and your brain in the Parkinsonian state says, well, I can't really handle all that information because it's all going through this rather tight network. Probably the best thing is to shut the network down, reset, rethink, let's move forward. And and how does dopamine fit into that? Because in some ways, if I give people dopamine drugs, as we do for Parkinson's, freezing of gait will actually really improve.
Yeah.
But my clinical experience is that doesn't always happen. So, chemical mediating this, or do you think it was something else that was doing it?
So I remember seeing Peter Jenner, whose name you'll remember and some people on the call will know, is one of the leading sort of neuropharmacologists who's helped us to develop new drugs for Parkinson's, mainly using what we call rodent models, where you can wipe out dopamine in the system and then test a new drug. And we asked, I remember somebody asking him the same question, which was, well, why doesn't, and basically Peter's response was very pithy and very accurate, which was, it's just a model. And
Yeah,
so, it's not the whole story. And I think the truth is, as we learn more and more about freezing, you can imagine a situation where you say, well, even at the best that we have with dopamine on board, people still have Parkinsonism, the slowness and it hasn't all gone away. So at those times of increased crisis, it's
yeah,
still going to fall over at times, right? And then on top of that, we're learning more and more about the fact that dopamine is not the only show in town. And people on this call may know, well, there are other neurotransmitters, so chemicals in the brain that allow us to do the things we do across the cholinergic nervous system with acetylcholine, which we often thought about as being, oh, that's important for memory. But it's also important for balance. It's also important for gait, and it's also important that those things are attention shifting, and then noradrenaline, serotonin, and we go, well, that's just mood or cognitive, and you go, no, no, no. Remember the monkey?
yeah,
It's going through the same pathway. We need to think about this and say, okay, well, if we were going to pick off a cure for freezing of gait, do we think there's one magic bullet that would do it all? The answer is probably not. Would it help most of the freezing in most of the patients? Well, that would be a good start.
yeah.
And obviously, we've got that with dopamine, but at the same time, it's far from perfect.
Okay. So those early studies you did with the imaging, you came to the conclusion that actually the hypothesis was correct, that there was a node where things were going wrong, and that was within the basal ganglia itself, or outside the basal ganglia, or was it
Yeah.
a whole network that seemed to be going a bit wrong?
Yeah, so I mean, I guess one of the big transitions in my, uh, if you like, neuroscience career in the last 15, 20 years has been the move from circuits to networks.
Okay.
And so, in terms of what we were
So what
able
do you
to do in the
mean by
virtual,
that? So for someone like
yeah,
me, I would say what a circuit is where things are connected to each other, and a network is where that is, but there might be two circuits. So it's just a bit more
yeah,
complicated circuit.
yeah. So, I mean, essentially, here we are, where you and I are a circuit. We're very clearly connected.
Yeah.
But at the same time, there are people all around the world at the moment in the States who you have never met, Roger, but you have influenced their life. There's no direct connection between you, but you're an influencer. So
Right.
essentially, we think about the circuit. You can look at it and go, "Well, I could cut that circuit. So let's block Roger Marker's emails, and we'll never talk again." But the network is much wider. So effectively, everything that your brain is doing is trying to get a more integrated pattern across different parts of your thinking parts of the brain. This is what the basal ganglia in part is really good at doing, which is to say, well, if I want you walking or if I want you swinging through the trees and looking for whatever, the fact of the matter is I need to get your arms moving, but also need to get your visual processing going at the same time so that you don't miss the snake and think that it's a vine and you grab the snake instead of the vine. And so the integration of the network is key. And so what we were able to do in the virtual reality was simple things first. We said, okay, well, what happens during freezing? Okay. What happens when people have to dual task, when they process the cognitive information at the same time as doing motor? What's the if you like the circuit that's involved and what's the network that's involved? Narrow doorways were in there. We've had patients in turning in the virtual reality, people walking over a plank so that they look like they're going to fall. And then you can tease out which parts of the circuitry are
Mm.
integrating the network. And of course, in terms of our treatment, what you'd really like is something that you could drop onto, let's say, the circuit and it would fix the network. The chances of us doing that with one clean magic bullet is tiny. And so we have to think about, okay, how do we support the network, perhaps by targeting various aspects of the circuit?
So basically, the research which you began with virtual reality and imaging has been the way forward. That's how you've continued to do the work. So a sort of simple question: how do you know that people are freezing in the scanner because they're not walking? So did they
Yeah,
sort of make a noise, say, "Oh, freezing," or
yeah,
because you're
that's
saying that's what you want to study, but how can you pick it up?
Yeah, so with the other demands, so you can actually compare sort of when they go through narrow doorways versus when they go through wide
Mm-hmm.
doorways, and then you just do a comparison. And you pick the periods when actually they're doing it okay without freezing. To look at freezing, what we can do is look at what's the output of the pedals and does it have a pattern? And then on top of that, what we were able to do is to get people into the clinic and actually get them walking around what we call timed up and go tasks to measure the amount of freezing they had in the real world, and then put them into a similar environment in the virtual world and correlated the amount of freezing those patients got. Now, walking on pedals, lying on your back in a scanner, rules out all of the balance processes. So it is, as Peter Jenner would say, just a model, but it's been very helpful in that sense. And it's allowed us to say, okay, it's not just in the basal ganglia that we're seeing these effects, it's actually affecting the networks. And then the question becomes, okay, well, that actually allows us some other approaches because um, we can use the functional imaging to look at the whole brain,
Mm.
the temporal resolution, that is to say, how quickly does the scanner respond to the brain's changes? It's relatively slow. But things like brainwave recordings, which you can then take, saying, well, we're expecting the network to impact the thinking parts of the brain that are more accessible if you put leads on the outside of the brain, or you have now the ability to record from deep within the brain, from some of the deep brain stimulators, from very particular parts of the circuit, which dictate the network. And also, I think someone in the chat mentioned
Yeah.
techniques like FNIRS, which, in very simple terms, most people will be familiar with a pulse oximeter where they measure your oxygen level by putting it on your finger. And essentially, in FNIRS, you can have a look at if you like, the pulse oximeter around certain parts of your brain. It doesn't cover the deeper structures, but that can give you clues. So I think that we have ways of modeling the freezing and then saying, okay, well, let's test the hypothesis and see if we can make it better.
So just so I've got the paradigm right, because I'm no expert in this and I've not done it, but I mean, so if I was walking around in your lab and you sort of say, well, I go through a door and I freeze, and that's pretty consistent, then on the virtual reality, would you see on their feet, on the pedals, that they sort of stutter? So
It's
and so you
yeah.
can actually see freezing in terms of the.
Sorry, Roger. I think you dropped out a bit on me there.
Yeah,
I
I was
was
just saying.
so the
So you
answer
can
is
actually
yes.
see this.
I won't share the screen now, but I have some nice video footage, and people may want to go to my website and have a look at some of these things. But effectively, we have video, and there's a phenomenon that we see with freezing in a fair percentage of the freezing episodes called trembling in place. And patients who experience freezing will often know this feeling where their feet stick, but one of them sort of keeps going, and it's
Yeah,
trying to go, as it were. And we can see that on the foot pedals. And interestingly, we have used the virtual reality paradigm in the operating theatre when patients are having deep brain stimulation done. So, as you know, Roger, patients are often awake during this procedure. And naturally, it's you need some very friendly neurosurgeons, but you can put a screen in the operating theatre. And while patients are awake, you can get them to do the virtual reality paradigm. And what we were able to show in that study was that when patients were freezing, we could get a bit of this trembling. We also saw a massive surge in that inhibition signal from deep in the brain. And when we looked at the muscles, so a muscle recording, an EMG, electrical myography from the legs, you could see that the movements were actually timed with these sort of false starts from the brain, trying to restart the system. So I think we can model the freezing phenomenon. Is it freezing of gait? Perhaps not entirely, but at the same time, there are lots of other freezing phenomena. So freezing with your hands, freezing of speech. I think they often share a
yeah.
similar common pathway.
I was going to ask you about that because we always talk about freezing of gait as though it really is only something that's specific for gait because it's the most obvious. But you're saying that freezing is you see it a lot with gait, as we say, but it actually is common to lots of other activities. So you have freezing of hand movements, freezing of speech, freezing of thought. Do you think because sometimes people say, "I really struggle to get things out."
Yeah, I've definitely got freezing of thought myself at times. This is true. And again, it's probably an overload of the system.
Yeah,
So freezing of upper limbs, which has a wonderful acronym of FOUL, freezing of upper limbs, we actually showed that. So the original virtual reality paradigm, I was trying to reduce head movement in the scanner, so I got people to use hand pedals. And the amount of freezing they reported correlated with the amount of freezing they got from hand pedals. And the reason we shifted to foot pedals is because Michael J. Fox, when they funded the research, said, "Oh, this doesn't really seem like freezing." And I was like, "Okay, you've got a foot fetish, that's fine. We'll do it with pedals, that's fine." And it's turned out okay. But so the answer is, I think there's a common pathway where freezing phenomena happens. And your question about freezing of thought is an interesting one. And one of the stories, that the anecdotes I have, is that in 2010, I went to the first workshop on freezing of gait that I'd been to. And I was going to present my new hypothesis and show people the data. And I remember walking into the breakfast area. So I was going through a doorway, and I could see as I came through the doorway, Stan Farn, the grandfather of the Movement Disorders and Parkinson's Society, bless him, who's well known to the WPC as one of the founders, talking to Mark Hallett, who tragically we lost at the end of last year, one of the greatest gentlemen I've ever met from the NIH, talking to Professor Strick, who'd done all the modeling of networks. And as I hit the doorway, in my anxious state, and saw, I froze at the doorway. I absolutely froze. And I thought, maybe there's something to this hypothesis. It possibly works. You just have to overload it enough.
yeah. So you've got this network dysfunction. So and this is something you're still working on to sort of better define it, I guess. But some of the questions which have come in, and also, one of the interesting aspects is how do you treat it? So the standard way you could give people dopamine and it works. These cholinesterase inhibitors, which I think most people know that rivastigmine drugs like this. People have tried deep brain stimulation. They're these sensory tricks and such like. So it would be useful just to hear what do you think are the best ways of treating it as a clinician, and then sort of layer that onto your model as to how it works? And actually, do you need to treat everyone in an individual way because they'll all respond differently to these treatments? So something about treatments, how it works in your model, and
Yeah.
what patient you'd recommend to patients.
So dopamine treatment obviously is up there. So we see most of the freezing related to dipping of the dopamine level. So getting that right is critical. Um, we should shout out to the physiotherapists. And
Yeah.
one of the things that people need to recognize is that gait, as we talked about earlier, is one of the complicated things we do, but you don't think about it. And so it's an automatic movement. And if I ask you to remember your driving test, when you went on your driving test, you were hands at 10 to 2, your head would turn to look at the mirror every 3 seconds, and you came out dripping with sweat. And the reason is because you're using your cortex. And gait, as well as driving, of course, as soon as you pass your driving test, you're like, one hand on the wheel, you're slumped, you never look in the mirror, you see a police car up in front, and everything comes back in, your attentional system kicks in again,
Yeah.
but it's too much energy to do that all the time. The brain uses sugar as its only energy source. It's too demanding to walk like that all the time. But what the physiotherapists are trying to do is to say, okay, I know you're used to walking as an automatic process, but I need you to think about it. I need you to make big movements. I need you to go from automatic to volitional control, and then try and make the volitional more automatic. So in actual fact, when the physiotherapists are trying to do this with patients, they're actually trying to shift your working load, as it were, so that you're using a bit more of the cortex, but you're not so worn out by it. So that's in there. I
Yeah.
mentioned the sensory tricks and the cueing. They tend to habituate. That is to say, walking over lines, it becomes tiring and draining. So I guess one of the things is, could we get people to do this as and when on demand? So, for example, we could talk about something simple first, which is to say, okay, well, if we had an EEG system, let's say it was just, I don't know, 4 electrodes in a headband that could 2 seconds or so before your feet freeze, recognize the signal in real time, and then send an alert to an earpiece with your wife's voice recorded saying, lift your knees, you're going to freeze, or something that you have on demand, rather than having that all the time. I've got to lift my knees, it's good. That might be a biofeedback that could work. And in a similar way, and this is one of the areas that's very hot at the moment, is could we use adaptive deep brain stimulation? That is to say, at the moment, when we talk about adaptive deep brain stimulation, we're trying to say, well, can we keep people switched on most of the time so that their dopamine levels aren't dropping? But if in real time, again, 2 seconds before there was going to be a freezing episode, could we tell the deep brain stimulation system to change the amount of current or the electrode contacts, and for that 2 seconds, abort a freeze, and then go back to the baseline? So that would be an on-demand system. And I think
So it
that's
sounds
going to
like about
be,
these sensory cues. So how do they work? Is it sort of, it somehow taps in a sort of subcortical way into your network, and it sort of short circuits it to pretend to be normal? How
so
are
the
they?
sensory cues, just so that people recognize what we're talking about here, we're generally talking about visual cues, so things like lines on the floor or footprints that have been cut out, or the classic auditory, metronome, tick, tick, and it can't be too fast. If they go too fast, their feet get shorter and then they freeze. The other ones would be vibrating shoes, and they're
Yeah.
usually a bit too late, so you've already frozen, and
And
then you
I
get the
don't know if you
sensory
have in
cue.
Australia. There's been quite in the UK they have these thing called Q1, which people wear on their chest, or wristbands. I don't know if they're something which many of
And then it
you.
vibrates and it
Yeah.
alerts people. So I think what happens is it allows a reset of the system. So the lines on the floor and the metronome, what you're doing is you're paying attention to your movement. So
Right.
what you're really doing is focusing on a volitional movement. The trouble is it's exhausting, and a
Yeah.
bit like that driving test example. The vibration things, they're basically a reset because you've already frozen by that stage. What you really want to do is to not freeze in the first
Yeah.
place. And I think, all of the things we're talking about, I think the deep brain stimulation, if it could work, that would be a really cool fix, because most people with Parkinson's, and most of us in general, don't want to have to do exercise all the time. We'd rather just take a tablet or have a surgery
Yeah.
and it'd be fixed. That'd be wonderful. And I think that's, one of the other thoughts around where we go with this. You mentioned the cholinesterase inhibitor story, and obviously Emily Henderson in Bristol has led the chief PD study, and I don't think it's reported formally yet. But the idea of using a neurotransmitter through the cholinergic nervous system to help gait, I think many of us have tried this with our patients, haven't found it all that exciting and interesting. Recently, my team, along with a couple of others, so Rad Wood, so Yorick Nonikers and Kaylina against Mertens in Waterloo, Canada, have won an award from the Michael J. Fox Foundation, which here in Australia will be supported by Shake It Up Foundation too, to look at modulating the noradrenergic system,
Yeah.
repurposing a medication called atomoxetine. And the noradrenergic system is an interesting thing because essentially, if you think about adrenaline, it's the thing that kind of cues you and focuses you. And so the idea being, well, if we could give someone if you like, a shot of noradrenaline that would help their focus, then that might reduce their freezing. And of course, give me a pill, doctor, rather than the exercise, I'll take the pill. So I think, these are one of the exciting areas we have to look at.
Okay, so just if you've got freezing, then physio is obviously a great thing. Some of these sensory cueing systems can help some people. Dopamine, as we've said, the cholinesterase inhibitors. Your opinion on those is that they probably don't do very much, or again, is it
I don't
individual
think,
shape? Because I've had a few people who really think it's worked quite well for them,
yeah.
but I don't
I
know
think
what
the
the
bottom line,
trial data really says overall.
yeah. So I think the bottom line is that, as with all of these things, there may be individual patients who do respond. So I think it's worth a try. And
Yeah.
of course, there are other indications. So poor cognition can respond to these drugs. Hallucinations, delusions can respond to these drugs. So there's a very low bar to saying, well, we should try this. I
Mm.
think that's perfectly fine. And the side effect profile is pretty low. The issue, of course, in countries can be that the drug may not be licensed. So then you
Yeah.
have to have somebody who's prepared to write the prescription and say, well, I'll take the risk on this. And I think you have to be aware of the fact that there is a massive placebo effect with most things Parkinsonian, where they get something and they'll say, "Oh yes, I am better now," or the caravan marches on, and the consultation you had 3 months ago, where freezing was the biggest problem, now it's all about their bladder or the hallucination, and they've forgotten all that on the day, doc. That was, and now the game has moved on, and so this is the variable feast of certainly advancing Parkinson's. We have so many things, like a firework display that keep going bang, 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 bang. What am I fixing next?
Okay, but atomoxetine is something. So this is noradrenaline, which has been associated with lots of things. Actually, I mean, depression is one thing that's been associated. But you think um, so? Targeting this system could be beneficial in its own right, not in combination with other drugs. It would just be if we can improve the levels of noradrenaline, this should make the system work in a more efficient way.
So it's about trying to integrate that network,
Yeah.
and saying, okay, if we had enough dopamine on board, but the network still isn't talking, can we raise the tide such that those things will actually start playing nicely together? So the study we're going to do is very short in the sense it's a single dose study, but we will tap into some of the functional imaging and saying, well, okay, if we look at the connections of those networks, do they get strengthened by a drug like atomoxetine? Would that give us confidence that we should do a longer drug trial? And one of the things that I will highlight is that again, courtesy of one of the leading figures that we lost recently, Nir Giladi, or Nir Giladi, I should pronounce it, from Tel Aviv, who we lost tragically way before his time, really championed and carried us such that we now have an international consortium for freezing of gait. And you'll love this, Rog, because the acronym, of course, is icy fog. So we're living in Cambridge where you have a lot of icy fog. You should be able
Yeah.
to remember that. And
Yeah.
so the icy fog has really done a great job. And if you're a researcher, we've just published, Nature Reviews Neurology, a new definition of freezing. So there's a clinical definition, there's a technical definition. And that may sound like small beer, but the bottom line is that if you're going to do a trial, you need to know that we're all scoring the same thing. And so one of the other things that's happening, again supported by Michael J. Fox, is that we are also piloting a test paradigm, which will be called the Giladi protocol. So that essentially within 5-10 minutes, we should be able to test or assess someone's freezing of gait in the clinic and then score it, and again take that to clinical trials. Because at the moment, you do a clinical trial and say, well, what's the outcome measure? You go, well, how much freezing? What's the definite? Okay, so the International Consortium of Freezing of Gait, thanks to Neil and his massive vision and leadership, and both losing Neil and Mark has been a very tough time for all of us in
Yeah.
neurology, definitely.
No, I mean,
But yeah, I think we're moving in the right direction.
Yeah. So yeah, I mean, it's sad we've lost such people who've made such a vast contribution in this area. So I want to sort of in the last 10 minutes just to pick up a couple of things. One is about your deep brain stimulation. So deep brain stimulation, you can obviously put the electrode in many different sites, and there was this view probably 15, 20 years ago. Everything's about 15, 20 years ago for me now. I don't quite know. Where you put the electrode not in the subthalamic nucleus, not in the globus pallidum, but you put it deep in the brainstem in the peduncular-pontine nucleus. And even though people didn't know quite what that was and whether we were all talking about the same thing, coming to your point about definition, and there was a lot of excitement that if we stimulated the so-called peduncular-pontine nucleus, the PPN, we could make gait better. And then the trial sort of slightly fizzled out, that it didn't actually really make much difference. So when you're talking about deep brain stimulation as a treatment for this, whether it be normal or adaptive, are we talking about normal sites? Are we going back to the PPN? And if we're going to different sites, what sites are we referring to?
I think it will probably be initially the standard sites.
Right.
So with the adaptive DBS at the moment, if you think about the concept, the idea is that you change the settings to keep people more on, so that like their dopamine levels dropping. So that would reduce the amount of freezing they get because of off. That sort of adaptive stuff is sort of over days rather than necessarily seconds. So one of the thoughts that I'm talking about is whether you could do it as a reactive system for a particular freezing episode. And here we've got data which says, well, actually, there are also studies looking specifically at freezing where if you could change not the contact points, but the frequency of the stimulation, so not necessarily the amount of stimulation, but how many times a second you're stimulating the brain, it seems to reduce freezing, but it doesn't necessarily last. But if you only had to do that for 2 or 3 seconds, 4 or 5 times a day, and then switch back to the normal frequency, then actually that might be an on-demand system that is a bit smarter. One of my big things that I like to champion is the concept of smart therapies. So I want to get away from advanced therapies or device-assisted therapies, because people have got the wrong idea about what that means. It means, well, my Parkinson's is very bad. No, you just need a smarter technology than a tablet that goes everywhere. So I think, I'm championing the term smart therapy. So when you're talking to your patients and saying, I think there's this thing called deep brain stimulation, there are these infusions, there's stem cell, they're smart therapies. Don't call them advanced therapies, because people get the wrong idea.
Oh dear, I'll have to change my nomenclature.
I think you should. I think you should. So Bas Bloom and I wrote this paper, I think last year or the year before. It was time to wind back the clock on smart therapies.
Okay. But basically the PPN is no longer.
I don't. Yeah. So the PPN, the trials that were done were very small and they were a little bit inconsistent. And I've got a wonderful video of patients who've had PPN who look fantastic with their stimulator on and terrible with their stimulator off, and they say, look, it's made no difference. So I think it's really interesting. It may well be that there are particular patients who are going to need different target sites, but I think most of the work at the moment will be looking not for new targets, but to say, okay, what can we do with the new technology that we have with our old targets that we know are safe?
And someone's asked about focused ultrasound, but presumably this would be less useful, because that's obviously where you make an actual lesion, with an ultrasound. And what you want is a system which is electrically active that you can modulate with the gait. So focused ultrasound probably wouldn't be useful for this type of problem.
No, and I think what I'd say about focused ultrasound is that the data for tremor is probably okay. But remember, you can only have one side of the brain done. Otherwise, the balance just gets terrible in these patients. And in terms of gait, it is this network. And so I must confess, I think if you're fit enough to have deep brain stimulation surgery, that's usually going to be a better option than the focused ultrasound.
So your experience has been people with DBS, actually their gait generally improves with DBS, or
so there's nice work out there from I think it's Schuppenbach, where they looked at the early STIM trial. So the early STIM trial said, okay, patients coming through who need to have deep brain stimulation because they start to get wearing off of dyskinesia. half of them are going to get best nodal therapy, half of them are going to get deep brain stimulation. And the study said, oh, their on periods are much better. But when they analyzed the post hoc analysis, what they also showed was that freezing was reduced in those patients who had the deep brain stimulation. And so I think there are all sorts of reasons. And I must confess, one of the other papers, which I'm hoping is going to get accepted soon, is I'm waging a war on people making cognitive deficits an exclusion for deep
Yeah.
brain stimulation. I have no idea why we do this.
Yeah,
So, I
but
mean,
also gait
essentially,
is said to be one. So people with bad gait, they're often
yeah.
excluded. You're actually saying this is actually
So I
wrong.
think
People
if you've got bad gait, if you've got bad gait and balance, that is an exclusion. But if you've got someone who's got a bit of freezing as part of their wearing off, then you say, well, that wearing off should improve, their
yeah.
freezing should improve. But if you've got someone who's falling and often falling backwards, you go, well, this isn't going to get better with deep brain stimulation. I should be clear on that.
And I sort of wanted to end up on another smart technology, to use the right phrasing, is, there's been a lot of interest in digital biomarkers, putting monitors on your axial muscles in your back, on your arms, to try and get a better impression of people's motor performance. And obviously, falls and freezing could be picked up by these devices. So how much is that featured in your research? How much is that in your clinical practice? And where do you see that fitting in going forward?
So I think that the wearables
Mm.
have found a place, and I suspect that they'll be used in future clinical trials. The concern is that there are a lot of things that can mimic freezing on the wearables. And I think that the technology, we've had that technology for over 15 years, probably 20
Mm.
years. And it really hasn't been a killer knockout blow that, oh, yeah, this wearable can detect freezing of gait with accuracy. I think it's getting better. And one of the technology, and I think it's really important because patients at home are very different to in the clinic.
Yeah.
So having that kind of data is really important. One of the things that's struck me is the sort of home radar stuff that's come out of Diana Katabi's work, where effectively using sort of a Wi-Fi signal at home, they've been able to show how gait might progress over, say, 6 months, and it's that leg rather than that leg. And whether you could adapt that technology and say, well, actually, you can now detect freezing because it's got a very characteristic pattern that's a bit different to just stopping on the spot. So I think those technologies might be able to help us with the real question, which is how do we improve people's quality of life? Because that doesn't happen for the half an hour they come to clinic, because then they're on their best behavior, and often because their adrenaline levels are through the roof. And actually, that's why they're probably a bit better.
Yeah. And also for your particular studies, they're in these nice big wide corridors with very few doors, and everything
That's
looks,
right. Yeah.
is a much easier place. So looking to the future, Summit, where do you think this work will go next? So where are you taking your work next? And what would be your ambition for, making a change in people's lives with this particular problem in the next sort of, I know we was talking 5-10 years, but I mean, it would just be useful to hear where you're going and
Yeah.
what impact do you think that will have?
So I think we've mentioned the idea of adaptive deep brain stimulation. So could we find a signal, change it in real time, stop you from freezing? And similarly, EEG, so ambulatory brainwave recordings, again with biofeedback that would tell you. We talked about modulating neurotransmitters like noradrenaline. So that'll be a study that we're calling the anti-freeze study, because of atomoxetine and freezing. So anti-freeze will hopefully start happening this year. And then one of the
That's
other
going to be a very
things,
strange concept in Australia, antifreeze, because they don't really.
absolutely, yeah. And then the other thing is we've had this virtual reality gait paradigm for ages, and so one of the things I want to trial is a thing called Gait Hero, where we actually let people take home the foot pedals and the paradigm and go train at home. So let's train you assess you in the clinic, send you home, go train, come back, because getting access to physiotherapy and actually committing. If we can gamify these things, that might be another approach that people might find, rewarding. And perhaps maybe we'll add the aliens back and we can start shooting them and make it much more interesting.
So really to take your technologies out into the real world with a
Yeah.
view that actually we will get a much better impression of what's actually happening in the real world.
Yeah.
And then using these combination of treatments to try and sort of work out how we can best improve what people can do.
Yeah. And the truth is we need to listen to the voice of the patient and the caregiver. I mean, the fact of
Yeah.
the matter is we may say people look really good, but actually, one of the things that we learned ages ago when we looked at cognitive impairment was we had like 2 hours of cognitive testing with the neuropsychologist and a questionnaire for the caregiver. And the caregiver was great at picking mild cognitive impairment. And the neuropsych, said, well, we don't need to pay you guys. We just ask the wife or the husband and they'll tell us.
Yeah. And so my final question, just before I conclude, is one of the things which has been very exciting in this field has been the initiative which you've been very heavily involved with about bringing the researchers together. So one of the criticisms we often get is it's great what Simon does in Sydney, but is he aware of what Baz is doing in the Netherlands? Are they aware of what Lynn Rochester's doing in Newcastle? How much do you talk to each other? So, I mean, you have brought this community together, so you're very much working together to try and solve this together. Would that be a fair? And that's something which has been very exciting, and I would think very catalyzing for the field.
It's beautiful, and again, call out to Near and to Mark as people that helped us to come together. I wrote Mark's obituary, and I said he was somebody who wanted to build collaboration, not empire. And I think what we see in the freezing community, particularly, is that everybody sort of knows each other and everyone's friendly, and we are working together more than, and I'm involved in a whole bunch of other consortia who don't work as well together, but the freezing consortia really are getting there and getting everyone together and across multiple angles. So I'm very excited about the future we have in that icy fog that we've established.
Well, I think we should bring things to a conclusion. I sorry that I occasionally slightly drop out here because I'm at home. I have to say also because I'm in some Ingmar Bergman film, which I keep looking at with this sort of light on my face and this dark top. But
I thought you were in the witness protection scheme.
Yeah,
and you got the lighting wrong.
But I mean, you've done amazing things, Simon. And I'm just very grateful to being part of this amazing story of yours and how you've taken on what was an incredible challenge. And you've made it something which is tractable and we can do something about. So I want to thank you very much for all you've done for the Parkinson's community in Australia, full stop, but particularly what you've done around your work, especially around Gait. I know it's not all that you do, but I mean, it's been great to see how that work has really developed. So thank you very much for that. I would like to thank again Supernus Pharmaceuticals for their support in making this series possible. The next one of these will be back at the normal time. So Simon will be up at 3 o'clock in the morning to watch this, which will be on March the 10th, where we're moving from the freezing that we've had today to Angela Chensi-Nielsen, who's a world expert on dyskinesias. So L-DOPA-induced dyskinesias, which she doesn't like to call them L-DOPA-induced, I think they're just called dyskinesias. So we're very much looking forward to that. That will be in March. And of course, 2 months after that, in May, we have the most important event of the entire year, which will be the WPC, the 7th one we've had, all the way from Washington in whenever it was 2006 now to Phoenix, Arizona. So do sign up for it. I'm delighted to say that we have had lots and lots of people coming to this meeting. We have lots of terrific people speaking, so do sign up for it. And all it leaves me to do is to thank you all very much for joining us tonight, to thank Eli for all the hard work she does making this happen, and particularly for Simon for giving us such an eloquent and beautiful story of how we're starting to unravel the problems of Gait. So thank you all very much.
Thank you, everyone. Have a great day.